Good morning, student. Today we will start the new topic, that is morulation and blastulation of frog. Morulation and blastulation of frog. After fertilization, zygote or embryo is formed. That cleavage process also completed, and after that morula and blastula, and after that gastrula. All this development of that embryo takes place with the different types of phases. Morulation and blastulation. The term morula has been defined as a globular solid mass, of the blastomeres. Blastomeres means that is the number of cells formed by cleavage of a zygote that typically precedes the blastula. Webster Adamstone F.V. defines morula as a stage in blastula formation in the mammals during which no cavity exists. Formation of a morula in frog and mammals. In frog, ovum is classed as a secondary microlithal and hence contains mere amount of yolk is homogeneous distribution, though mammals have originated from the reptiles with heavily yolked eggs. Megalithical, their ova and cleavage pattern following closely like those of primitive aquatic chordates like branchiostoma. Their eggs undergoes the equal holoblastic cleavage, which for all practical purposes is an unmodified mitosis. The study of cleavage divisions in mammalian eggs was first carried out in the pig ovum by Acetone, Schreiter and Husser as the deutoplasmic matter is scanty. The first cleavage furrow passes through animal pole. Animal pole is the anterior side and vegetal pole is the posterior side of that egg. Animal pole and vegetal pole axis and cuts the ovum into two equal blastomeres. The second cleavage division takes place at right angle to the first. Further cleavage divisions occurs in the close succession until a small group of blastomeres is formed with the zona pellucida. A thick capsule with radial striations enclosing the mammalian ovum. This developing embryo with a spherical ball of cells is called as morula. This developing embryo with a spherical ball of cell is called as morula. Morula, that is the morula stage after cleavage. Lewis and Hartmann made an extensive study in the eggs macaque monkeys and secured a complete series of cleavage stages, Hartwig and Rock obtained similar results in the human ova. Blastulation. As the cleavage proceeds further, the number of blastomeres increases and they undergo a profound rearrangement. The addition of the blastomeres to each other increases and they arrange themselves into a single cell thick ciliates echinoderms, ciliates and echinoderms, and cephalochordates, etc. All these animals, that is the invertebrate animals, or many cell thick, most vertebrates. True epithelium called the blastoderm. True epithelium called the blastoderm due to the rearrangements of blastomeres to form a blastoderm. A fluid filled space or cavity called the blastocele appears almost in the center of the blastoderm. Blastocele means that is the cavity. This cavity at first appears just by narrow crevices between the blastoderm, but gradually increases in the center during the rearrangement of cells. This hollow, spherical and uniepithelial thick embryonic stage. Uni means single, epithelial thick embryonic stage is called as blastula, and the process of its formation is called as the blastulation. In the blastula, specialized cell adhesions appears in the most peripheral layer of the cell and a true cell sheet is formed. As cells cleave, they are integrated into a physiological continuum by tight junctions, desmosomes and cytoplasmic bridges. Tight junctions appears at apical ends of epithelial cells. And organize a coherent sheet. The epithelial layer surrounds the developing embryo and controls the exchange of materials between the environment and the blastocele. Desmosomes gradually appears in embryonic tissues as specialization of the cell surface. For example, in the early thick embryo, we find no desmosomes, but we do see tight junctions. The tissues are easily dissociated at these stages, but in older embryos, when tissues are more highly organized and distinct, desmosomes are found at numerous points in plasma membranes, and the tissue are much more difficult to disintegrate. The desmosomes and tight junctions are uncommon in invertebrate tissues. In their place are present separate desmosomes with similar adhesive functions. Process of blastulation. How the process takes place in the blastulation? How does the first embryonic cell sit, the blastoderm develop? This question has been studied in the hollow sphere blastula of the echinoderms, where the hyaline layer at the cell periphery plays an important part in the blastula formation. The hyaline layer is the extracellular matrix of the cleavage embryo. The following two theories have suggested to explain the mechanism of blastulation. Two theories suggested. Number one, hydrostatic pressure theory. This theory was suggested by K. Don in 1960, and according to it, the hydrostatic pressure within the blastocele forces the cleaving cells to periphery. According to this view, fine cytoplasmic process penetrating the enveloping hyaline layer, creating a cohesive expanding surface. As cell number increases and cell becomes smaller, they secrete fluids into the extracellular spaces, and these fluids accumulate in the center cavity. Hydrostatic pressure builds up in the cavity, collides with outermost cells adhesive to the hyaline layer and generate a hollow spheres. Second number, that is surface tension theory. First is hydrostatic pressure theory, second is surface tension theory. This theory was forwarded by Gustafsson and Walpert. In 1967. According to this theory, the formation and expansion of blastocele results from the manner in which the blastomeres are packed in a blastula just before each cell divides. The surface tension of the outer cell layer increases and the cells round up, divide, and produce two rounded cells. Since cleavages are synchronous, this produces rhythmic tensions, and divisions are translated into pulsations of contraction and expansion. After a division, tension is reduced and cell contact is increased. Blastulation in isoleucithal egg. In microleucithal and isoleucithal egg of Amphioxus, the blastomeres at the end of cleavage arrange themselves into the single cell thick simple columnar epithelium enclosing the centrally located blastocele. These cells are not for exactly equal size due to slight Yock inhibition of vegetal cells. Therefore, at the vegetal pole, the epithelial is thicker, and at the animal pole, it is thinner, thus the polarity of the egg persists in the polarity of the blastula. Second is blastulation in moderately telolicital eggs. Types of eggs आहे, that is the yolk material present in the egg, telolicital at the tail region, isolicital, mesolicital. This is the yolk material is present in the egg, and that yolk is the food for the embryo. Blastulation in moderately telolicital eggs. In moderately telolicital eggs such as frog and other amphibians, the yolk has an unequal distribution. This results in the displacement of the horizontal cleavage towards the animal pole and retardation of cleavage rate in the yolk laden vegetal half. As a consequence, the blastomeres of vegetal half macromeres are much larger than those of the animal half micromeres. The small micromeres tends to form the roof of the blastocele, which the large macromeres form the floor of blastocele. Third is blastulation in heavily telolicital eggs. In the egg of fishes, reptiles and birds, at the end of segmentation, the embryo has arrived at the morula stage, which consists of a disc-shaped mass cell lining applying to the yolk. In the latter stage of cleavage, the blastomeres of the center area of blastodisc become separated from the underlying yolk and enclosed between itself and the uncleavage residue of egg, a fluid-filled cavity, subgerminal cavity. Okay, thank you.